Hello, everyone, and welcome to today's webinar, Antibody Engineering for Therapeutics and Research Tools. I am Cassie Soltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Vector Laboratories. To learn more, visit vectorlabs.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. Questions submitted will be answered by our speakers after the presentation. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. I'd like to now welcome our speakers for today's webinar. Dr. Michael Fiebig, Chief Scientific Officer at Absolute Antibody, a Vector Labs company, and Dr. Vera Molkenthin, Director at the Antibody Unit at 2Bind GmbH. Michael, you may now begin your portion of the presentation.
Well, thank you very much, Cassie, for the lovely introduction. And just before I get going, I'd like to thank Patty and Chengya from the Absolute Antibody and Vector Labs marketing team for all their help and effort in getting this webinar set up. Thank you, Patty. Thank you, Chengya. Now, I'm Michael Fiebig. I'm the CSO of Absolute Antibody, a Vector Labs company. And today I want to tell a little bit about our experiences in applying antibody engineering strategies, not only to the development of therapeutic antibodies, but also to research tools. And I'll take a little dive also into raw materials used for IVD. To understand how we've ended up working in this area, I'm going to have to tell you a little bit more about Absolute Antibody first. Slide changes, good. Now, we're an antibody reagent and service provider exclusively focused on recombinant antibody technology. And we're founded with a vision and the mission that everyone across biomedical research should have access to recombinant and engineered antibodies, not just folks working in the pharmaceutical space. As part of that, we offer an ever-growing catalog of recombinant antibodies, as well as a portfolio of recombinant antibody services, including antibody sequencing, antibody engineering, which includes multi-specific antibodies and humanization, as well as transient expression and subsequent purification. We were founded in 2012, originally in Oxford, but soon moved our labs to the Wilton Centre in the ever-sunny northeast of England. In the summer of 2024, we merged with Vector Laboratories, further expanding our joint service offering into specialty chemicals and bioconjugation services, as highlighted here on the bottom right, with a nod to our biodesign toolbox. Now, this slide really sums up the core of what we do at Absolute Antibody. Once you have the sequences of an antibody, and you know the sequences of the variable domains that mediate antigen binding, you can convert this antibody into virtually any format to suit your experimental needs. These can be naturally occurring isotypes, like in the first three rows here, or these can be artificial designs, such as recombinant fragment or multispecific antibodies. Now, what I'm showing here, when you're grafting the antigen binding domains of an antibody onto a UFC domain, is called chimerization. I will talk a little bit more about the difference between chimerization and humanization later, but it's important to note that the first therapeutic antibodies were made by chimerization. So taking, say, a mouse or rat or antigen binding domain and putting those onto a human IgG1 Fc domain. These kinds of therapeutics can quite readily be spotted because they have the syllable Xi or Ximab in their name. So things like infliximab or rituximab or cetuximab. Now, outside of the therapeutic antibody space, we've always believed in the utility of this for the research reagents market, even for fairly simple things like co-labeling studies, where you just wanted a little bit more diversity of primary antibody species to allow for selective secondary antibody labeling. But what I want to show you in the following are some examples of where we're pretty much supercharging the utility of clones through chimerization. When you're working, say, in mice, a lot of the antibodies that are being used in vivo are rat or hamster antibodies. Now, these antibodies, once you inject them a couple of times, they become immunogenic and the mice start feeling quite poorly and they lose efficacy. And likewise, a lot of these antibodies are in the isotype they came out of the hybridoma fusion. They might not necessarily be in the isotype that's actually desirable for the experiment you're trying to do. And now in human therapeutics, this has been overcome by chimerizing the antibodies, so putting them onto human Fc domains so they're less immunogenic, and then choosing whether you want a depleting or non-depleting Fc domain. We've spent a lot of time expanding a portfolio of anti-mouse antibodies that we've engineered to be more suitable for in vivo applications. And pretty much as shown here, swapping the Fc domain around and then putting the desired Fc domain on there. And one particular model that's really been taking off of late is an antibody that's directed to Ly6G called 1A8. That was originally a rat IgG2A, so quite a poorly depleting antibody, which we've changed over to a mouse IgG2A. And this group around Olofsson, they were using it to develop a really robust neutrophil depleting model that's been used quite widely now. And this model shows, and this antibody in this model shows vastly superior activity to the original rat antibody, while still being really well tolerated. So this is an example about the use of a chimeric IgG, but what about non-IgG isotypes? These can be important analytes in serology tests in areas such as autoimmunity, allergy, or infectious disease. We can, of course, take any antibody and turn them into human IgEs, IgAs, IgMs. And in fact, it doesn't even need to stop at this. We can quite readily do this for livestock, companion animals, or for animals that form natural reservoirs of infectious diseases. During the COVID pandemic, a panel of human IgG1, IgA, and IgM antibodies based on an anti-SARS-CoV-2 antibody we made was listed in the WHO manual for the preparation of reference materials for use as secondary standards in antibody testing. I'm sure you can imagine that was quite a special moment for the team. And I want to take just a couple minutes to dive a little bit deeper into this kind of application. When you're making these standards and controls for serology tests, generally your calibrators and controls are based on patient-derived material. This can be hard to obtain sometimes, and there can be biohazard concerns associated with this, especially the nastier the infectious disease agent gets. Also, these can often be finite. Just can't get that much material, and in order to overcome this, these are often pools, which does reduce variability, but also sort of smoothens out the contents of those. And they're kind of undefined. You know, if you ask what is a representative COVID-19 serum, it might not necessarily be a mix of hundreds of different samples pooled together. And so going back to the concept that we can take any antibody and turn it into any isotype, we started making these controls that were precisely defined blends of IgG1, IgMs, and IgAs, etc. And these also work if you actually put them onto antibody tests, as shown here on the right-hand side. Now, you can say, are these really representative of human of a human serology sample? And the answer is no. But are they standardized? Yes, they are standardized. And so once you have absolute definition of your control and your calibrator, you can get absolute quantification, which a lot of people made use of to really benchmark their tests against other tests. And there's some really nice studies out there really looking at how well different tests compared once you actually had an absolute sample. Now, this was on sort of naturally occurring isotypes. What if we looked at those non-natural constructs that I had on the bottom row of the previous diagram, and that sort of populate this periodic table of antibody formats that we prepared a while back? These are all antibody formats that we have made for clients of ours, and there are even some more now. A lot of these formats, a lot of these complex biologics are made with the aim of making anti-cancer drugs at the moment. But this field is also branching out into autoimmune diseases and into infectious disease and even neurological disorders. And so it's an exciting space to be working in, and it's exciting also from a protein engineering point of view. But it's not easy. Complex biologics also means often complex workflows. Now, we do have experience with well over 200 antibody formats, and with that also comes a need for experience with lots of different protein engineering solutions. We quite regularly work with off-patent as well as proprietary solutions for things like light chain pairing, heavy chain heterodimerization, and we also play around a lot with modulating FC functions. So whether it's just silencing the FC or giving the FC's specific affinity for FC receptors, as well as half-life extensions through modulating of the FcRn binding site. Now, expressing these molecules is just part of the challenge. You also still need to purify these because they always make some byproducts. And so we have quite a wide range of purification strategies available to really tailor those to the needs of the specific molecule, and likewise quite a battery of characterization methods available to really ensure that what we're delivering and what we're promising in terms of high purity and low endotoxin levels is met. These complex biologics, however, they don't just stop in the therapeutic space. A lot of people want to take some of those mechanisms back into the basic research space. And this can be just to study the underlying biology of the immune response, but also to explore whether or not those could be used in novel immunomodulatory approaches. Now, human antibodies, as you can imagine, once you put them into a mouse, they are immunogenic after a time. They are transgenic in humanized mice, but these come with some caveats, especially around immune cell activation, and they can be quite expensive. So we decided to embark on a mission to generate these syngeneic, fully murine bispecifics to create surrogate molecules comparable to human bispecifics. Initially, we started off with the kinds of molecules shown on the bottom left, where you're just appending the FC domain with binders. But then I think where we truly cracked the nut is when we developed a murine knob into whole FC heterodimerization platform that allows us to build these asymmetric molecules shown in the middle. And on those, we can then start building and even make tri-specific antibodies and what have you. And just as a little side note, we've also not stopped at mouse. We've also got a fully canine platform available for folks working in the veterinary space, and that can be quite translational work, actually. So let's look at a little case study for the use of one of our mouse bispecifics. This was a study where researchers were using a bispecific antibody that targeted a tumor cell and targeted CD3 on a T cell. To recruit the T cell to the tumor cell and mediate killing. Now, this wasn't just any old T cell. This was actually T cell that was induced towards a virus. So it's actually a virus-specific T cell. And what they did with that was to essentially take a cold tumor that wasn't showing a lot of T cells in it, induce T cells in the periphery with an oncolytic virus, and then pull those T cells into the tumor and recruit them to the tumor cells using these bispecific antibodies. Essentially, taking a cold tumor and turning it into a, well, let's call it a reheated tumor. This was just a really nice example of what you can do once you have these kinds of basic research tools available to you. Now, humanization. I mentioned chimerization already, which is sort of a step from going from a mouse antibody towards something more human. And you can see we've got the mouse antibody fully on the top right. We've got the chimeric antibody we've got the mouse antibody at the top left, we've got the chimeric antibody one position to the right, and then we've got the human antibody fully in blue on the right. But in between that, we've got the humanized antibody, where we've taken the antigen binding loops, so just these very short stretches within the variable domain, and grafted them onto an acceptor framework. Now, this process has been around for quite a while now, and the idea was always that these antibodies will be a lot less immunogenic than chimeric antibodies or mouse antibodies. Now, a while back, we looked at data of actually clinically observed immunogenicity, and we actually weren't able to find a really, really good correlation between either percentage identity to the original to the closest human germline, or if you're starting to predict immunogenic peptides. You see a difference between fully human antibodies to humanized or chimeric antibodies, but the difference between chimeric and humanized antibodies wasn't really falling out as much as I think we might intuitively hope. What we did see with all of these is there are always outliers, and there's definitely always biology that further contributes to immunogenicity. However, regulators do pretty much expect you to humanize an antibody nowadays if you're trying to build a therapeutic. Even though last year there was actually a chimeric antibody that went into the clinic, so it's not a full stop. However, we thought, hey, so if you already have to humanize your antibody, what else can we give you for your antibody project to make the molecule even just better, even if maybe the immunogenicity concept is not as stark as people expect. And we decided on focusing on manufacturability of the antibody. Your best antibody is useless if you can't make it at scale, and your best antibody is useless if it keeps on aggregating on you. And we definitely know that aggregation is an important driver for immunogenicity. So we launched a service called Prometheus, which delivers a panel of humanized antibodies. And it has a unique focus on enhancing antibody manufacturability by increasing expression, lowering aggregation, and improving long-term stability. It is a process based on CDR grafting, but what we do is we have a uniquely curated cohort of acceptor frameworks. So here we've gone through lots and lots of data that's out there on manufacturability and stability of acceptor frameworks, and already threw out all the acceptor frameworks that are likely to misbehave, so we don't have to fix that later. And of course, we also backmutate some non-CDR residues to optimize affinity. Now, this service does guarantee a variant that's comparable in activity to the parent antibody. And we can start from mouse and rats. And now that we've launched Prometheus 2.0, we can also start from rabbit and camelid antibodies. And we've completed over 230 humanization projects in the last 8 years. And just to show a little bit what I mean with improving yields and aggregation profile, on the top right here, you see this is some actual data from a case study. You've got the little black bar on the left, that is the input antibody, the chimeric version of that. You see fairly low relative expression, and then which, as shown on the left-hand side, y-axis, and then a definite propensity for aggregation as shown on the left-hand side y-axis. And pretty much all the variants we generated using our method had better expression levels and a better aggregation profile. And when we then took this also into some functional testing, you've got IC50 value on the y-axis and relative expression and expression relative to the chimeric antibody on the x-axis. We could find a really nice cohort of antibodies that performed as well, if not even a tiny bit better than the original. I think that's due to better folding, and they expressed massively better than the original antibody. Now, with this, you can sort of ask, well, what makes a good antibody? And because a lot of the characteristics that you might want of an antibody are universally desired. So let's take for therapeutics, for example. As I said, we want an antibody that has high expression levels. We want an antibody that shows low aggregation. We want an antibody that's got long-term stability and low long-term and low immunogenicity, even though we can put this little asterisk behind that as I described earlier. Now, for diagnostics and research tools, what are we after? Well, actually kind of the same things. I mean, we might not need the low immunogenicity because unless you're maybe trying to control for hammer response, but we just want the good manufacturability. And so it's probably not going to come as much of a surprise to you that we're working with quite a lot of diagnostic companies to help them improve manufacturability of their antibodies without fundamentally changing the binding characteristics of their antibodies. At the same time, similar approaches can be applied to research reagents as well, and to components of research reagents. And I'm going to show you an example of the latter first, looking at how individual components can be optimized. This harks back a little bit to the complex biologics story I told earlier. Now, single-chain FVs are important components in lots of constructs that people are making, things like bispecifics, but also CAR-Ts, and scFv-based drugs and imaging agents. Now, these single-chain FVs, they're literally the variable domain of the light chain linked with the variable domain of the heavy chain with just a little linker, and that's it. Mouse, rat, or hamster antibodies really struggle to reliably convert into single-chain FVs. Human antibodies, on the other hand, seem to have a much better conversion rate. So when we're tasked with something like this, we quite often fall back on either full humanization or partial humanization to, with generous back mutations, to screen for ways of converting a mouse or hamster or rat antibodies to single-chain FVs. An example for this was actually an anti-CD3 antibody that we used as part of our mouse bispecifics campaign. That was it's a fairly well-known hamster antibody that just would not convert. And by turning it, by partially humanizing it, we're able to create this really, really well-folded single-chain FV that is doing all the heavy lifting in this construct. Now, this example is an example of how we made better mouse antibodies with our humanization platform. Now, where am I going with this? We started off with a rat antibody. That's a well-known antibody targeting mouse monocyte markers, but it is known for really strong immunogenicity. And even with chimerization to mouse IgG2A, it was just a poor expressor. Initially, we thought, well, the rat and the mouse, they're pretty similar. We just need to swap out a couple of amino acids and solve the problem. But unfortunately, this killed expression altogether. We then decided, let's take our humanization approach. Let's find the human framework that we would normally use, and then find the closest mouse homologue to that framework. Kind of thinking, if we can get a well-behaved human framework, maybe we can now find a well-behaved mouse framework. And we did this, and we were able to graft the CDRs onto a new acceptor framework. We got 8-fold increased expression levels, and we even got slightly better activity by flow cytometry. You see no binding to neutrophils in the left-hand column, and improved binding to monocytes in the right-hand column, with the bottom version being our engineered version. And again, I think this is around tight folding that might have made the antibody a little bit better. And once we put this in vivo, because this was an antibody commonly used in vivo, we actually see much deeper depletion, even with smaller antibody doses and slower recovery, which indicates this antibody is able to stay active for a lot longer. But there is still some optimization and progress on this. Now, summary for my part, I hope I've been able to show you that antibody engineering is not just for therapeutics. Many antibody-based tools and raw materials can be improved with it. And therapeutics development strategies inspire superior research tools and raw materials. I mean, we all want the same thing. We want reproducible, functionally optimized, and excellent manufacturability characteristics. And if it's good enough for therapeutics, it's good enough for research tools and IVD, I think. There are lots of different workflows available to tailor your antibodies to your assays. And The biology dictates the structure you need, and the structure dictates the workflow that we need in order to get to these molecules. And finally, this is not the end of our developments. With the capabilities across the whole Vector Labs organization, we're now in a position to explore more opportunities beyond just the antibody protein itself, especially in the form of advanced bioconjugation methods and specialty linkers. There's some first successes in this space that I hope I can speak to you about in the future, but I feel we're only starting to scratch on the surface of what's possible, and it's a really exciting space to be in. Now, that's all I want to tell you about at this point, so thank you very much for your attention. I will gladly answer some questions later, but first I want to pass the word to Vera from TwoBind. I'm really glad she could join us today, because with all this talk of good antibodies, at some point you're actually going to need some methods to prove good biophysical characteristics of your antibodies. And this is where good friends at TwoBind come into play. So without further ado, Vera, will you take over?
Yes. Thanks, Michael, for the kind introduction, and thanks to Vector Laboratories for having me here as a guest speaker. So I'm happy to share some of the exciting results with you that we had with a joint project together with Absolute Antibody. But before I start with the project, I would like to say a few words about TwoBind. So TwoBind is a German CRO, and we support antibody development. And I'm quoting Benjamin Franklin here: an investment in knowledge always pays the best interest. And we believe this sentence is very true when you intend to develop a therapeutic drug. And it is even more true if your therapeutic drug is a bispecific antibody. So we can support you to answer all questions around the interactions of your molecule. And that includes affinity, avidity, specificity, and stoichiometry. It also includes real-time kinetics on cells. And actually, that's a new technology that bridges a long-time existing gap between the SPR, KDs, and the EC50 values that you determine in flow cytometry. In addition to that, we have detailed looks at conformational and colloidal stabilities. And all this data should enable you to take informed decisions, do your risk assessment, and develop appropriate mitigation strategies. So the project was actually a stability study with bispecifics. And Absolute Antibody provided us with high quality material for all the nice formats that you can see on the right side of the slide. And all these molecules are part of a anti-HER2, anti-CD3 bispecific reference panel. And the beauty of this set is actually that all these molecules share the same variable domains. So they use the trastuzumab variable domains to target HER2 and the OKT3 variable domains to target CD3. As you can see here, the molecules have different formats. The set included formats with FC parts and formats without FC parts, and they had the single chains fused to different sides of the molecule. Since the only difference between these molecules is really the format, this is actually an ideal set to study the influence of the bispecifics format on the stability of the molecules. To do so, we use a multi-parametric Panther assay, and that actually enables a simultaneous measurement of the unfolding and the aggregation in the thermal gradient. So we use the nano-DSF signal to monitor the unfolding of the molecule, light scattering and back reflection to monitor the aggregation. It delivers high-resolution data to detect subtle stability differences between different candidates, and it also enables a domain-specific profiling. So we do see individual transitions for the unfolding of individual domains. It enables a label-free operation, so we don't have to add external dyes or labels. We use the intrinsic protein fluorescence. It's a very efficient workflow, so we can analyze up to 24 samples and duplicates per run, and we need just 10 micrograms of material per sample. This high throughput and low material consumption enables the placement within the lead selection workflow. So we can compare different candidates for their unfolding and aggregation behavior. But of course, we can also use the technology at a later stage to check the desired candidate in the different buffer conditions, how the buffer conditions influence the thermal stability and aggregation behavior. So to set the frames, I would like to give some background on unfolding, aggregation, oligomerization. And if we start with the lower part, the purple color on this slide, so this is actually what we are monitoring in the thermal gradient in the Panther experiment. So we start with the folded native state. We heat it up and monitor the unfolding with the nano-DSF signal. In addition to that, we also monitor the aggregate formation. And to do that, we use the light scattering for the smaller aggregates and the turbidity back reflection measurement for larger aggregates. Typically, we start with an isothermal DLS upfront just to check the material before we heat it up. So we use DLS to assess the colloidal status, and we use the hydrodynamic radius and the polydispersity as readout factors. So even a fully folded antibody can form oligomers, and that is a property that is typically called self-interaction. So some antibodies have the tendency to bind identical molecules and form oligomers. This can be an existing equilibrium, or it can also lead to aggregation in the long run. So let's have a look at the material that Absolute Antibody provided to us. So you see the molecules on the right side of the slide, and on the left side, you see the intensity distribution that we obtained in the DLS. And you can see that all the colors show a very sharp and single peak, and there's no sign for aggregation, which would show up at higher hydrodynamic radii. The cumulant PDI was below 0.1 for all of the samples, and that confirms a highly monodispersed sample quality. The hydrodynamic radii were different between the molecules and correlated with the molecular weight of the constructs. So we were in a very good starting situation, and as a next step, we heated up the molecules. And this is what you can see here, just for a selection. It was a really huge data set, so I had to choose some comparisons here, and can't show you everything. And in this comparison, I have an IgG, the trastuzumab, and 3 Fc-free formats, the tandem single chain and 2 bispecific Fab single chain fusion proteins. And on the left side, you see the data. So on the x-axis, you have the temperature, and the different panels show the different readouts. So on the top panel, we have the first derivative of the unfolding curve. So it's not the unfolding curve itself, it's the first derivative, and transitions show up as peaks in this plot. To monitor the unfolding, we use the nano-DSF signal, and in particular, the ratio between 350 nanometer and 330 nanometer. And in the middle plot, you can see the aggregate formation in the thermal gradient. For this, we use the turbidity signal measured by back reflection. And in the lower plot, you can see the hydrodynamic radius measured in DLS, all within the thermal gradient. And comparing the IgG, which is in red, and the 3 Fc-free formats, you can see that actually the first transition is kind of at the same temperature. So all the IP1 temperatures are around 70 degrees, no major difference here. There is a difference between the tandem single chain and the full-length IgG, and that's the missing second transition. The single chain has only one transition, the IgG1 has two transitions. And if we look at the aggregation behavior, we see actually a huge difference. Yeah, so the IgG starts to aggregate at 10 degrees higher temperature compared to the FC-free formats. And all the FC-free formats show a reduced colloidal stability to the full-length IgG. And actually, you wouldn't have guessed that from just seeing the unfolding curve. It is confirmed by the increase in the hydrodynamic radius. So also in this case, we see an earlier start for the FC-free formats compared to the IgG. And overall, the increase in the cumulant radius starts at even lower temperatures because we can detect smaller aggregates or smaller size increase oligomers already. Okay, so that's the influence of the FC on the colloidal stability. So FC helps to keep the molecule in solution to keep to retain the colloidal stability. And on the next slide, I'm showing you another comparison, and that's different bispecific that have the single chain fusion at different sites of the molecule. Yeah, so it's more or less a regular IgG, and single chain fragments are fused either to the C terminus of heavy or light chain or to the N terminus of heavy or light chain. And here in green and blue color, we have the C-terminal fusions. And again, the IP1 looks nearly same, so not a major difference on the thermal unfolding behavior, on the first transition at least. And in the middle plot, you see the aggregation behavior. And here we can see that the reddish colors show an earlier aggregation compared to the blue and the green colors. So N-terminal fusions have a reduced colloidal stability compared to the C-terminal fusions. And if we compare the two N-terminal fusions, we even see a slight disadvantage of N-terminal fusions to the light chain. So this one here on the right lower corner, the single chain fused at the N-terminus of the light chain had the lowest aggregation temperature in this comparison. So coming to the summary. So if you allow me some general advice, so please consider the thermal stabilities when you choose your bispecific formats. There can be differences, even if you use the same variable domains. We highly recommend the multiparametric Panther assay. It enables domain-specific insights, and it also enables the parallel monitoring of the unfolding and the aggregation behavior. So as you have seen, the unfolding and the aggregation temperature can be different. So it's really important to look at both of these parameters. And some specific findings for the HER2 CD3 bispecific reference panel. We saw that FC domains increase colloidal stabilities. That's not too surprising, actually. I mean, FC fusions are very common for the production of recombinant proteins because they increase the solubility. The second finding is maybe a little bit more unexpected. So we saw that it made a difference where you exactly fuse the single chain. Yeah, so we saw that C-terminal fusions are preferred over the N-terminal fusions, and the N-terminal fusions to the heavy chain are preferred over fusions to the light chain. So, and with that, I've reached the end, and I think we are ready for Q&A now.
Wonderful. Thank you both for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just type your question into the Ask a Question box and click Submit. We'll answer as many of your questions as we have time for. Our first question is for you, Michael. This question's asking, for someone that is new to making bispecifics, what is the process to identify the best format?
Ooh, that's a good one. We can have very, very long conversations about that. I think first it's important to take a step back from the format and really think about the biology that you're trying to tackle. What are you trying to engage with? Are you, say, trying to pull an immune effector cell to a tumor, or are you trying to and then what kind of effector cell could that be? If it's a T cell, are you holding on to CD3? Maybe that's a monovalent engagement you want to form. If you're trying to engage with natural killer cells, maybe you want to do that a little bit differently. And then if you're trying to target, say, a tumor cell, do you have a situation where you can make use of avidity and sort of maybe have multiple binding sites towards a specific target to make use of targeting tissues with higher overexpression of a marker versus tissues that might just naturally express a specific antigen. And once you've sort of plotted out that a little bit, then you can start looking at all the possible designs there are. And then maybe you might also consider IP considerations, whether or not specific methods, specific solutions are available in an IP-free space, or whether you might just want to explore all the possibilities that are out there. There is no ideal format, but you sort of have to sort of slowly creep up on it, and sometimes in an iterative way. But I'll gladly follow up with you after the call, and maybe we can have after this webinar, maybe we can have a discussion around the precise situation that you're trying to address.
Wonderful, thank you. Our next question here is for you, Vera. This question is saying, in your example, it appears as if not every unfolding event leads to immediate aggregation. How do you explain this observation?
Yeah, wow, that's a hard one also. Yeah, so actually, some of the unfolding is reversible. So it's not that every unfolding immediately causes the aggregation. It can actually go back to the folded state, and some molecules even have this behavior to show an equilibrium between some partially unfolded domains and the fully folded format. And as long as it doesn't lead to aggregation, it can coexist even. And the Panther has also the possibility to test for reversible unfolding. So that's something that is possible. We haven't done it in that cases although.
Excellent, thank you. Our next question, we're going back over to you, Michael. This question is asking, what formats do you recommend starting when making bispecifics?
Yeah, I think this ties in a little bit with the question we had earlier. The kind of biology you're trying to achieve does give you some inputs, but if there's not a lot known about your system, I would start off with an architecture that has a 1-to-1 ratio of binding site for each target, 2-to-1, sort of an asymmetrical, an antibody with an asymmetrical valency, and then a 2-to-2 construct. These kinds of molecules can already give you an indicator for which arrangements give you the desired mode of action, and then you can start refining on that. The more you know about the biology of the system, the more you can probably fast track this process. But 1-to-1, 2-to-1, and a 2-to-2. Sometimes you can't get around those.
Excellent, thank you. Okay, back over to you, Vera. This next question's asking, what is the advantage of nano DSF over Cipro-Orange assays for measuring thermal stabilities?
Yeah, so the first advantage is you don't have to add external dyes. Yeah, so adding the Cipro-Orange as a monitor for the unfolding can actually cause artifacts and yeah, just cover things that are really ongoing in your molecule. The second thing is that the nano DSF has a better resolution compared to the Cipro-Orange assay. So you can see more individual transitions in the nano DSF compared to the Cipro-Orange assay. And then, yeah, the third thing is if you combine the nano DSF with the light scattering technologies or back reflection, you can also monitor the aggregation in parallel.
Wonderful, thank you. Okay, Michael, next one is up for you. How do I know if the engineered antibody will express well?
Sorry, this is going to sound a little bit flippant, but by making it, we're still not in a place where we can reliably predict how well something will express. We can probably flag some elements and say the variable domains if they don't look right. If an antibody doesn't have the right elements in the framework sequences, if cysteines aren't in the right place, that usually means that something is going to be a little bit problematic. But if all those patterns are in place, you can still get a lot of variability. So sometimes, well, you generally just have to go and make the antibody and learn from that. And that, unfortunately, is biology for you sometimes.
Okay, perfect. Thank you. Okay, our next one is for you as well, Michael. This one is asking, if the antibody is not going to be used in vivo, why would I want to humanize it?
Very good question. You don't want to normally you wouldn't want to humanize it. The example that I alluded to, or the examples that I alluded to, where we're using humanization to improve where we're using humanization for diagnostics companies that are not using it in vivo but in vitro assays. This was entirely because the humanization approach that we use improves manufacturability characteristics. So it's not important that the antibody at the end is humanized. It's important that the antibody at the end of it has high expression levels and low aggregation potential. So it's kind of a byproduct from the process that we're using. And no, they don't need a humanized antibody in an in vitro assay. They just need a well-behaved antibody in their in vitro assay.
Okay, great. Thank you. All right, our next question here, I'm not sure which of you, so just grab it if it's for you, but what type of linker do you recommend between the different domains on a bispecific or trispecific antibody?
I think this one's for me. It depends. It depends where the linker is. In a lot of cases, we just use 4GS linkers or copies of that and that's between domains of let's say if you're making single chain FEs, we use 4GS linkers. If you're appending something to the C terminus, we tend to use 4GS linkers. Now, if you're putting things above the hinge, you can make use of quite a bit of sort of natural linker real estate there, and so make use of those upper hinge residues. We have at times used other linker sequences, sometimes with the aim of adjusting isoelectric points a little bit, so putting some charged residues in there if we're having some solubility issues. But I have to admit that generally we start off on a 4GS linker. But that again, if that's something that you're working on and perhaps struggling with in a specific context, maybe we can follow up afterwards and look at the specific molecules you're working on. Thanks for the great question.
Wonderful, thank you. All right, our next question, we're going to jump over to you, Vera. This one is saying, once identified stable bispecific target structure, would different VH and VL sequence substitution, or different clone, would that change the characteristics, stability, and functionality?
Yeah, so I believe so, yes. So in most cases, the variable domains will influence the stability. And I would recommend also to have a look at if the variable domains are stable as Fab and single chain fragment as you want to introduce them into your bispecific. So the relevant domain format, that is something that could be useful to check upfront. So it could give you an idea whether it's, one sequence is principally usable in that particular format or not. But overall, it's something that has to be tested, actually. Yeah, the variable domains can definitely influence the stability of the bispecifics. Yes.
Wonderful, thank you, Vera. Okay, our next question, this one is for Michael. Have you tried generating rabbit-human chimeric antibody?
Yes, we actually make quite a lot of those. I suspect you might be asking because rabbits have 2 kappa light chains, and one of those, the kappa, the K1 light chain, has an additional cysteine that is just before the cysteine, before the light chain CDR3, and this cysteine forms an additional disulfide bond with another cysteine on the light chain kappa 1 constant domain. So if you've asked this question, you might have already worried about this at some point. And it is possible to form rabbit-human chimeric antibodies. Sometimes you need to address the presence of that K1 cysteine, and sometimes you don't. We have worked on methods where we've just put it on and it worked. And if it didn't work, then we've gone back and either mutated the position in the rabbit light chain, or even looked at altering a human kappa chain as well. For the other kappa K2 and lambda light chains for rabbit, we've not really seen any issues. So by and large, these work quite well, and rabbit antibodies tend to be good to work with, if you bear in mind that they've got this little idiosyncrasy hidden in one of the kappa chains.
Excellent, thank you. And it looks like we have one more question here for today's webinar. If you have any additional questions, get them in now. We'll try to answer as many as we have in the last couple of minutes. This question is asking, if more unstable at 70 degrees Celsius from turbidity or DLS assays, what does that mean in real life for the stability of the therapeutic, or where storage would mainly be at negative 20 degrees Celsius, 4 degrees Celsius, or injected at 37 degrees Celsius?
Yeah, that's a wonderful question. So thanks for that. It's actually, of course, we are all hoping that nobody would try to heat up an antibody to 70 degrees before injecting it into anybody. But it's a kind of a predictor. So if you rank different molecules and you see stability limitations at elevated temperature, that's a kind of an accelerator. So if you store it for a long time at 4 degrees, then it might, the one that shows the lower stability at 70 degrees might degrade earlier. So that's the rationale behind that. Yeah. And also, it's not only the IP1, it's also very important to consider when it actually starts. So the T on, the onset of the unfolding, and also the onset of the aggregation. So if that is close to room temperature or close to 37 degrees, that's of course more worrying even. Yeah.
Okay, excellent. Thank you, Vera. Our next question, we did have a couple come in. So this next one is asking, is the extra disulfide bond in rabbit IgG causing trouble in humanization?
Very good follow-on question to what I was saying about the K1 light chain in rabbit. It requires extra attention. It does not cause trouble as such. It's just something to bear in mind when you're evaluating activity after the humanization process. If for some reason, whichever path you've chosen, either to remove the cysteine or to leave the cysteine in and tweak the human constant domain, if all of a sudden you're seeing funny behavior there, then it might tell you to go the other route. But we've been really successful with rabbit antibodies, and so we're perfectly happy to take those on as well. So it's something to bear in mind. Definitely keep an eye out for it.
Okay, excellent. Thank you. And we'll squeeze just one more question in here today. This last question is asking, in the process of evaluating the 230 plus SCFVS, did you observe any common reasons for instability like misfolding or aggregation? I'm trying to understand how screening helps eliminate such issues.
I think in general, when you're talking about single chain FVs, you have to be mindful of the propensity of them to form aggregates. I mean, even if you get them to express in the first place, they do this thing where they sort of breathe open and then shut again, and and then refold again. And if they're in the vicinity of other single chain FVs, they can form these dimers and larger aggregates. So this is in a way a misfolding, this breathing open and closed, leading to aggregation. We haven't seen any, at least I've not been able to really identify any elements that make this better or worse. It's again sometimes just a question of making them and seeing how they behave. And quite often you can already glean a lot from a simple HPLC, sec-HPLC plot, and take it from there. But even if you purify them, even if you purify just a monofraction, you can start refolding, you can start reforming aggregates from this. So yeah, CFVs can be a bit tricky, and maybe in the future there will be tools to better predict which ones are better before you get to making them.
Wonderful, thank you. I want to thank you both for your time today and your important research. We would also like to thank LabRoots and our sponsor, Vector Laboratories, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions submitted during the live and on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.
Thank you. Goodbye.
Goodbye.